All right, good afternoon, everyone, and welcome to today's NAVBO webinar sponsored by the Education Committee. I'm Amber Stratman from Washington University in St. Louis, and I'm moderating today's session. So I'm pleased to welcome our speakers, Dr. Dionna Kasper from Dartmouth College and Dr. Jason Fish from Toronto General Research Hospital. Before we begin their sessions, I'd like to remind you that all NAVBO activities are now easily found in the calendar of events on our website. Please be sure to register for some of these upcoming events. Throughout each presentation, please type your questions into the question box, and those will be answered at the end of each talk. This webinar is being recorded and archived on the NAVBO website for future use if you want to go back and watch anything. So I'm very pleased to introduce our first speaker, Dr. Dionna Kasper, who is an assistant professor of molecular and systems biology in the Gesell School of Medicine at Dartmouth College. Dionna has a long-standing interest in uncovering the molecular mechanisms that govern tissue formation. Her graduate work using the C. elegans model discovered one of the first transcription factors that controls expression of PIWI interacting RNAs, so piRNAs, a germline-specific non-coding RNA that's essential for fertility. She then went on to investigate the functions of microRNAs in blood vessel development in the zebrafish during her postdoctoral training with Dr. Stefania Nicoli at Yale School of Medicine. Discovering that microRNA-mediated regulation shapes how endothelial cell surface proteins are post-translationally modified with specific N-glycans, leading to the groundbreaking finding that the N-glycan is a key endothelial cell determinant of hematopoietic cell fate. So at Dartmouth, the Kasper lab uses a combination of live imaging, genetics, and biochemical approaches in the zebrafish to dissect how N-glycosylation and other gene regulatory mechanisms control endothelial cell fate decisions. So I think we're excited to hear a lot about the work her new lab is doing. And so welcome, Dr. Kasper. I'll let you take it away.
Thanks, Amber. It was a great introduction. So yeah, I'm Dionna Kasper. I'm a fairly new assistant professor at the Dartmouth Gesell School of Medicine. Today, I'm going to talk about a lot of my published work that was when I was a postdoc in Stefania Nicoli's lab at the Yale Cardiovascular Center. And then kind of finish up the talk talking about how that work has really started up my own laboratory at Dartmouth. And so today I'm going to talk about how I identified a new regulatory mechanism that regulates the endothelial-to-hematopoietic transition. So a major biomedical goal in our field is to have the ability to gene correct and reprogram patient-derived cells into hematopoietic stem and progenitor cells, or HSPCs, in order to then reintroduce into patients who may have different blood disorders or leukemia. But unfortunately, we lack a robust way to mass produce hematopoietic stem cells in vitro that can then have normal lineage potential and can mass expand and graft for a long time. And so this suggests that we still lack an understanding of some of the key hematopoietic stem cell specification mechanisms. My lab, as well as others, try to identify these elusive mechanisms by studying how hematopoietic stem and progenitor cells arise naturally in vivo. Depicted here is in zebrafish, but that similar things happen in other organisms like mouse and human. And so the way hematopoietic stem cells originate is only during development and during a narrow window during development. And this is the only time where you can generate your lifetime pool of hematopoietic stem and progenitors cells. And this happens via an endothelial to hematopoietic transition, or EHT. And so during this process, a subset of specialized cells within the floor of the artery called hemogenic endothelial cells gain hematopoietic potential through kind of dramatic changes in gene expression where we're downregulating endothelial gene expression and increasing hematopoietic stem and progenitor gene expression. And these cells then round up and become hematopoietic stem cells entering circulation. And from there, these cells will then enter downstream hematopoietic organs such as the marrow and differentiate into all different blood cell types. And so the heroic work from many labs in the field have identified numerous factors that are important for regulating this transition. But we still lack an understanding of how are these different pathways regulated through time and space to undergo this dramatic cell identity change. And so the way I like to think about this is how are endothelial cells coordinating this fate change? And maybe one way to do this is to thinking about what global mechanisms might be regulating this complex gene expression changes. And so that could involve epigenetic, post-transcriptional, and post-translational mechanisms. And this is something that my lab is very interested in understanding. And today I'll tell you a story about how I went from looking at some post-transcriptional regulation that then informed post-translational mechanisms, and so bring the idea that there could be multiple layers of regulatory mechanisms. In addition, a question that we're interested in is, if you miscoordinate or disrupt this complicated process, are there functional consequences to adapting the wrong fate? So for instance, if you have too many or too few blood cells, blood stem cells, can you affect the downstream differentiation of those cells into different various lineages? And so when I started in Stefania's lab, I was inspired by my small RNA biology background from graduate work, and really wanted to understand how post-transcriptional regulation by microRNAs, if that was one of these global mechanisms that could regulate vascular cell fate. So microRNAs are small non-coding RNAs that imperfectly or bind through imperfect complementarity through target mRNAs and cause their degradation and translational repression. And these microRNAs have this really dramatic or this remarkable gene regulatory capacity, because one microRNA can affect hundreds and downregulate hundreds of different targets. So they can affect multiple effectors, but then you can also have a group of microRNAs that then target a whole many components of a signaling pathway. And so this seemed like a potential pathway that could regulate vascular development. And so in the NICLE lab, I carried out a phenotypic screen in which we looked to see if there were defects in hematopoietic stem cell specification and other cardiovascular defects in zebrafish mutants that had global deletion of microRNAs that we found were either highly expressed or enriched in endothelial cells that we FACS sorted from these animals throughout different major developmental stages. And so we came upon a really interesting microRNA, MIR223, and this microRNA we had found through an in situ hybridization type screen that it had an increase in the number of cells expressing CMIB, which is a marker of hematopoietic stem cells. And so because this microRNA was expressed in endothelial cells, but affecting hematopoietic stem cells, we really wanted to understand whether this was affecting the endothel's hematopoietic transition. And so to do this, we first wanted to understand, get a better idea of where MIR223 was expressed. And so to do this, we created a transgenic GAL4 UAS reporter where we integrated GAL4 into the MIR223 promoter. And when coupled with UAS GFP, could mark MIR223 expressing cells in green, and then coupled that with a vascular reporter in mCherry. And then analyzed during that developmental time stage. Here you're looking at a lateral view of the zebrafish embryo. And remarkably, we found found cells that were exhibiting behaviors of EHT. So these cells were in a salt and pepper fashion. They displayed very flat to budding morphologies and were primarily localized within the floor of the dorsal aorta where EHT occurs. We also imaged these cells and did see them kind of rounding up and undergoing processes reminiscent of EHT. So you can see right here, these cells are now going to bud off of the dorsal aorta. And so kind of to confirm that these cells were indeed cells that like hemogenic endothelial cells undergoing EHT, we performed single cell RNA sequencing of M-cherry positive endothelial cells at 27 hours. And so here you're looking at the developmental trajectory of those cells. And then we identified which cells are expressing miR223 based on the presence of the GFP transcript, which you see in blue. And as expected, or confirming our cell biological data, we could see that miR223 was expressed within the EHT trajectory. And so we could see some cells in the aortic endothelium, but was primarily within the clusters that represented the hemogenic endothelial cells, cluster 8, as well as cells that were hematopoietic stem cells primed for different lineages. And specifically, we found miR223 primarily or highly expressed within the population that was primed to form lymphoid and myeloid type cells. And so this gave us confidence that it was expressed in the correct cell types. And so we went on to then address whether it was required for endothelial-to-hematopoietic transition. And so we looked at various markers for the EHT trend or the EHT cell types. And so what you're going to see here is the vasculature in red and various markers for hematopoietic stem cells in green, and we generally are counting the number of cells that are undergoing the transition. And so from this analysis, GATA2B marks hemogenic endothelial cells, and from this analysis we could we found various phenotypes, but namely we found an increase in the number of hemogenic endothelial cells, an increase in c-MYB positive HSPCs, which was suggestive of an increase in the specification of these cell types. But we also saw changes in the behavior of different hematopoietic stem cell markers. And so we found an increase in HSPC proliferation using an EDU staining. So we saw an increase in the number of, or the increase in proliferation of cells marked with c-MYB and KDRL, but not their neighboring endothelial cells that lacked c-MYB expression. And so it was this increased proliferation was specific to the transition. We also had quantified how long these cells take to delaminate from the wall, and had found that in the mutants, we found an increase in budding time. And so from there, we, not shown here, but saw that throughout later stages, we could see an expansion of these hematopoietic stem cells at the later hematopoietic sites, such as the marrow and the thymus. And so we showed that this microRNA was important for regulating the transition, but also wanted to address there's more hematopoietic stem cells. Is there a consequence to having more? Maybe it is just a good thing to have more and that you can make different blood cell types, or is there a negative consequence? And so we looked at differentiation, doing various different stainings, and actually found skewed differentiation toward the lymphoid and myeloid cell lineages. So in the mutant background, we could find an increase in T lymphocytes that are shown here as marked by RAG1 expression in the thymus in the zebrafish at 6 days post-fertilization, as well as an increase in the number of neutrophils at 6 days marked by Sudan black staining. Whereas data not shown here showed little effect on the erythroid population. And so what we could find here is that this correlated well with the expression of miR223 affecting the hematopoietic stem and progenitor population. We've also found that this lineage differences had to do with expression in endothelial. It's due to its function in endothelial cells, and that if you overexpress miR223 specifically in endothelial cells, you could rescue the differentiation defects. And so this kind of led us to the model that miR223 was one of the first inhibitors identified of the endothelial-to-hematopoietic transition, and specifically was preventing the transition that generate hematopoietic stem cells that are primed for the lymphoid and myeloid lineages. And where it got really interesting for the story, besides identifying a new inhibitor, was asking or identifying how miR223 was limiting EHT. And so we wanted to find what its direct targets were. We had performed comparative transcriptome profiling and had narrowed in on a pathway that involved glycotransferases. Specifically, we had done additional sensor assays to show that ALT2, monosyltransferase, and ST3-GAL2, acyltransferase, were direct miR223's direct targets. And so to show that these were involved in the process, we used a guide RNA to mutate the binding site and showed that phenocopied the miR223 mutant when we prevented miR223 from binding these specific transcripts. So this showed that this interaction was important for that. So these transferases are important for putting for glycosylating proteins. This is a post-translational modification, specifically N-glycans. These are big sugar chains that are put on N or asparagine residues of proteins. And this is just how they're represented in the field, with each shape and color representing a specific sugar. And so like other post-translational modifications, they are important for establishing protein folding, stability, and activity. And so we thought this was interesting that we now have layers of regulatory mechanisms seeming to impact this process. And so we went on to ask whether the glycome was actually affected in these animals. And so we isolated endothelial cells, and then in collaboration with the Labria lab at UC Davis, isolated the glycans in endothelial cells to see if there was a difference between wild type and miR223 mutant. And there was a dramatic change where we saw a huge reduction of glycan subtypes that were terminally monosylated or had less mannose, and then had a dramatic increase in sialic acid, and that's represented here. And we could show that these target genes, if we overexpress them in endothelial cells, it could phenocopy miR223, kind of confirming that pathway. What's interesting is that we then went on to identify which proteins might have this signature of glycosylation, and we found really interesting candidate list. And these proteins, all but except integrin beta 1b, have not been implicated in the endothelial to hematopoietic transition. But what's interesting is that in other contexts, they often regulate factors that we do know are involved in EHT, just they themselves haven't been implicated. So we had things involved in EHT signaling, blood differentiation, proliferation, and cell adhesion matrix. And so now we have this really great candidate list that my lab is currently following up on and identifying new regulators of this process. And so we found a new mechanism in which a specialized sugar code within endothelial cells determines how these cells are undergoing the endothelial to hematopoietic transition, and that's governed by miR223 regulation of the N-glycome. And so when you don't have miR223, you have an upregulation of these genes, changing the glycome and then promoting production of lymphoid myeloid primed HSCs. And so the open questions my lab is addressing are, now that we know that this new pathway is involved, what are the cellular functions of glycoproteins? And so I have, we're following up on a lot of these different proteins, and are they regulated? And but more importantly, how or why do we need N-glycosylation to regulate their activity? And then finally, how do these proteins interact with already known EHT regulators? And I just want to give you a little flavor of some promising data. So one of our most interesting candidates is ADAM10. ADAM10 is important for regulating the processing of Notch signaling. And so ADAM10 is the first enzyme that cleaves the Notch receptor once ligand is bound, and that's followed by gamma secretase, and to then release the Notch intercellular domain to promote Notch gene expression. And so our hypothesis right now is that potentially glycosylation-dependent ADAM10 activity might be necessary to regulate Notch dynamics during EHT, because during EHT, Notch is highly regulated throughout this transition process, needing to be high, low, and then high again. What we found so far is that in our mutants, ADAM10 activity is reduced, and that if we overexpress wild type or a version of ADAM10 that lacks the glycosylation, that we get more hematopoietic stem cells. And so we are working now with the model that in the MIR223 mutant, when we have ADAM10, which is glycan deficient, that we're getting more cells because it's reducing, or Notch activity is even lower. And so we're now testing this hypothesis by looking at different cell types as well as measuring Notch activity. And just a final slide, I want to leave you with the impression that the glycan code might not just be for the endothelial-to-hematopoietic transition, but it might be a more global regulator of cell fate decisions or other ones. And so there's another cell fate decision, the endothelial-to-lymphatic transition. And from our single cell data, if we analyze Go terms from the cluster representing the vascular or the lymphatic and venous cluster, we have evidence of other overrepresented terms focusing on the glycosylation pathway. And so it'd be interesting to follow up to see if there is a glycan code and certain sugar modifications are important for these different cell fate decisions. So I wanted to thank my new team, various members who are working on different aspects of that. We have a fun time together. Also, I'd like to thank the Nickley Lab for really establishing a great program for me to work on in my new lab, as well as various collaborators and funding sources. And I'm happy to take any questions.
Excellent. Thank you so much, Dionna. That was great. Great talk. So let me get the questions pulled up here. We have a question that says, I might have missed this part. I was wondering how the two MIR223 targets how the two targets were identified in this study.
Yes, thanks for that question. I didn't get a chance to go over it, but we compared through bulk RNA sequencing wild type and MIR223 mutant endothelial cells, and then we identified which genes were upregulated in the mutant and then contained a MIR223 binding site. And from there, we were able to do different sensor assays and other experiments that then showed that they were directly targeted. And so that's kind of how we initially pulled those genes out. And then I think kind of the key evidence that I showed there was that if we can disrupt the binding site in the 3' UTR of those genes, that we could see the same phenotype that we were seeing in the mutant.
Yeah, very cool. I'm really compelled by this this data you showed at the end, that the glycome may be regulating cell fate choices, and in a broader context, do you see, maybe you're just starting down this path, but do you see differences between arteries and veins? Do you have an idea how you think that might be regulated?
Yeah, so it's really interesting right now. What we know is sort of at the gene expression level, just kind of this early data that from the single cell. So there's different glycosylation enzymes. So it's not ALG2 and ST3-GAL2, but other enzymes that would suggest other modifications. So these enzymes put on very specific modifications. So we're finding additional. So they're within the process, there's hundreds of these different types of enzymes. And so this is a different pool. So I would love for in my lab to do exactly what you're saying, is to probably fact sort out these different cell populations and then do the profiling for the glycomics. So the data I originally showed was just all of endothelial cells. Would be great to do artery, vein, lymphatic cells, hematopoietic stem cells, and see if we can actually work out a specific code.
Sure, that's very cool. Next question is wonderful talk. There's 2 questions here. So how does Notch link to EHT? And does MIR223 also have an impact on arterial specification?
Okay, yeah, those are great questions. So from some of the analysis that we looked at, we didn't see a change in markers that regulate arterial development. So we believe it's acting directly on the hemogenic population. Plus we see MIR223 kind of in that same kind of salt and pepper pattern of only some of the arterial cells do that. And so we believe it's just working at the blood level. And then, sorry, the first question was?
Was how does Notch link to EHT?
Yeah, I kind of had to go through it a little quickly, but we know Notch is really important where you need different levels, and a lot of times Notch activity, the levels of Notch activity are important for whether you need arterial specification, which is kind of why you need it high here, and then later down you still need the Notch signaling, but at a lower level for the hemogenic population, and then later for maintenance of the hematopoietic stem cells. And signaling through Notch establishes certain or stimulates the expression of different regulators downstream of that are important for this process. So Notch signaling binds different promoters and promotes their expression.
Cool.
But I think Notch is acting at a lot of different spots within here.
Yeah.
It'll be exciting to figure out where this regulation could might be playing in each of these. But we think it will be kind of more in this hemogenic population, but we still need to test that.
Very cool. We have 2 more quick questions, and I think they're both really important. So I want to give you 1 minute to answer each, to answer them. The first one is, you see this transition happen during development. Do you also see changes in this transition due to age? And then the second question is whether looking at N-glycan marks can be a good indicator of having EHT or not.
Yeah, so I think we're trying to figure that. So the EHT is only happening during development, but people are trying to be able to recapitulate this in a dish, and there's definitely models where you can use induced pluripotent stem cells and differentiate, and that would be the idea to eventually get it into clinic. But we don't see evidence of these different of EHT in adulthood, at least to my knowledge. But in terms of finding the different markers, I think it would be great. We have some evidence that if you use different drugs that say promote silation, that we treat embryos, we can increase hematopoietic stem cells. So this kind of gives the idea that if we profile them and understand the marks, that then we can then be using chemical ways to then recapitulate that same type of code in say these in vitro models, and that maybe we can help bypass some of these things. So I think it'd be great and something probably long term I would like to establish this type of model and chemical approach in the lab.
Very cool. Well, thank you, Dionna. We really appreciate your presentation today. If you have additional questions for her, feel free to drop them in the chat and we can send them to her after the talk complete today, and she'll email you back directly. So it's now my pleasure to introduce our second speaker, Dr. Jason Fish, who is a senior scientist at the Toronto General Hospital Research Institute. He received his PhD from the University of Toronto under the supervision of Dr. Philip Marsden, and completed his postdoctoral training at the Gladstone Institute of Cardiovascular Disease at the University of California, San Francisco, under the supervision of Deepak Srinivasa. The Fish Lab seeks to understand the contribution of endothelial cells to health and disease. In particular, the lab is deciphering how endothelial cells respond to physiologic and pathologic changes in the microenvironment via changes in gene expression. So the lab has identified important roles of non-coding RNAs, such as microRNAs, in modulating signaling pathways in endothelial cells. They have also identified signaling pathways that are involved in vascular development and vascular malformations, including the recent discovery that active KRAS mutations are present in the endothelium of the majority of sporadic cerebral arteriovenous malformations. And so we're excited to have Dr. Fish here today with us, and I will let you take it away.
All right, thanks, thanks, Amber. Can you see my screen all right?
We can.
Okay, excellent. All right, so I'll get started.
There's your bar at the bottom is there.
Yeah, I don't know how to get rid of that. Oh, there we go. Okay.
Perfect.
Right. Great. So, thanks for the introduction, and I'm happy to share some of our ongoing work that is trying to understand how somatic activating KRAS mutations are driving brain arteriovenous malformations. So brain AVMs are these abnormal connections that happen when an artery directly connects to a vein without going through a capillary network. And as you can see from this schematic here, these abnormal vascular structures can become remodeled over time. They form a really complicated nidus with tortuous vessels. And these are prone to rupture, and actually they're the leading cause of hemorrhagic stroke in young people. The majority of these are actually sporadic with no family history. And up until several years ago, there was no idea about what the genetic cause of these malformations might be. We now know that KRAS mutations drive the majority of these sporadic brain AVMs, but we still really don't understand how KRAS is causing these malformations. So far, the treatment is primarily surgical, and this comes with high risk for these types of surgeries. And so we'd really like to be able to go towards pharmacological treatments, but we really need to fully understand the molecular mechanisms to be able to implement this. So this schematic from Mika Vikula's group really shows the progress in this field of vascular malformations. There's a large number of different vascular malformations. Many of these are inherited, and in the early 90s, a number of mutations were discovered in inherited forms of vascular malformations, such as hereditary hemorrhagic telangiectasia and cerebral cavernous malformations. And these were typically loss of function mutations, so that individuals inherited one faulty copy of these genes. What was found over the ensuing decades was that within the lesion themselves, there's often a second hit and a loss of the wild type gene for those loss of function alleles. Really a watershed moment in the field was in 2009 when Mika Vikula and his team found activating TIE2 mutations in venous malformations, which were sporadic malformations. And since then, there's really been a very large number of different sporadic vascular malformations where there are somatic activating mutations in the endothelium. And I'll tell you more about brain AVMs and the discovery of KRAS activating mutations. So our lab started working in this field through a collaboration with Ivan Radovanovic, a clinician scientist at Toronto Western Hospital. And he operates on a number of patients with brain AVMs, and he was collecting samples during surgery and conducting whole exome sequencing of the brain AVM tissue and matching blood to determine whether the mutations that he found were truly sporadic mutations and not germline mutations. What he found was recurrent mutations in the KRAS gene, including G12V and G12D. These are activating mutations that are quite common in cancer, but they'd not been found in endothelial cells previously. We were able to confirm these activating KRAS mutations in 2 additional cohorts and found that the majority of patients had activating KRAS mutations. This included G12V, G12D, and Q61H. And these are all very common mutations in cancer. Interestingly, the variant allele frequency was quite variable. With some vascular malformations with quite low numbers of mutant KRAS cells, suggesting that you don't need to have all 100% KRAS mutant cells to form a brain AVM. And we found that the mutations were exclusively found within the endothelium. KRAS is a really important effector of cellular signaling. And in a normal cell, a receptor signal will signal through KRAS and activate a cellular response. And this is a very dynamic process where KRAS toggles between an active form that's GTP bound form to an inactive GDP bound form. In the setting of transformed cells where there are these KRAS mutations similar to the mutations that we found, it really gets locked into an active form that constitutively signals. And in cancer cells, this can signal through PI3 kinase, the MAPK-MEK-ERK pathway. Interestingly, what we found in brain AVMs is that mutant KRAS seems to signal preferentially through the MAPK-MEK-ERK pathway. You can see here phospho-ERK staining in the normal brain is very, very low. But in an AVM, you can see strong signal in the endothelium as well as other cells within the lesion. And if you isolate endothelial cells from the normal brain or the lesion, you can see that phosphorylation of ERK indicates preferential activation of the MAPK-MEK-ERK pathway, but not the PI3 kinase pathway or the MAPK-P38 pathway. In some of our initial characterization of cells expressing mutant KRAS, we found that electroporation with a G12V construct together with LifeAct GFP to label the actin cytoskeleton showed that these mutant cells are very much larger than normal cells. And this is in a confluent monolayer. And what we found is that these cells are very active. They dynamically rearrange these lamellipodia and as you can see from the cell traces, move quite actively even in a confluent monolayer. Ruin Wu in the lab has developed some tools to further look at the mechanisms of what KRAS might be doing to drive pathology. And he's developed stable DOX-inducible constructs where he can express an M-scarlet tagged wild type KRAS or an M-scarlet tagged mutant KRAS. In looking at some of the cellular phenotypes, he finds that in the absence of DOX induction, this is in a scratch assay, the cells don't fully close the wound, but activation of mutant KRAS, the cells are much faster in their migration. And this is a MEK-dependent phenotype. If you add a MEK inhibitor like SL327, the migration is inhibited. Similarly, if you do a spheroid sprouting assay, induction of KRAS leads to a dramatic amount of sprouting, and inhibition of MEK can suppress this. And one of the other phenotypes that we see very consistently is the disassembly of adherens junctions. And this makes sense considering the images that I showed you before, that these cells are very actively migrating and constantly rearranging and moving even in a confluent monolayer. So if you express wild-type KRAS, there's really no change in V-cadherin staining, but induction of G12V leads to disassembly of the adherens junctions, as you can see here with V-cadherin staining, and these cells have quite high levels of MEK-ERK signaling. This phenotype is very much MEK dependent. So you can see here disassembly of the junctions with activation of KRAS, but inhibition of MEK can restore this. And another phenotype that we see in conjunction with this is dysregulation of the extracellular matrix. And this really confirms something that George Davis published recently, showing that activation of KRAS can lead to degradation of ECM proteins, including fibronectin. You can see quite dramatic decrease in fibronectin in these underneath these cells, and this is restored again by MEK inhibition. And you would anticipate that with this degree of disruption of cell-cell junctions as well as matrix, these monolayers would be leaky. And indeed, that is the case. If we did a transwell leak assay, we found that expression of mutant KRAS led to increased FITC leak across the barrier. And this was restored with 2 different MEK inhibitors, but a PI3 kinase inhibitor had no effect, again, showing that it really is this MEK pathway that's driving these phenotypic changes. The degradation of the ECM was interesting to us, and we wondered whether mutant KRAS cells might be influencing their wild-type neighbors. As I mentioned before, AVMs are a mix of both wild-type and mutant cells. And so we really want to understand some of the non-cell autonomous effects of KRAS. So what we did in this experiment is mix together our inducible system with M-scarlet tagged wild-type or mutant KRAS, and mixing this together with wild-type KRAS cells that were labeled with F-tractin, which is a very bright red label. What we found in these cell mixing experiments is if we induced wild-type KRAS, or we used the G12 line without induction, fibronectin staining was very normal. But you can see when you activate mutant KRAS, which is in this region here, which has a dimmer M-scarlet tag, there is a significant downregulation of fibronectin. And what we found quite intriguing is that there are regions that are occupied by wild-type cells where there is significant degradation of fibronectin, suggesting that this is a non-autonomous effect, and that mutant KRAS cells might be affecting the phenotype of wild-type cells. We also use these cells to develop an AVM on a chip, and this work was driven by Dr. Sarah Vasconcelos' lab. She's developed this model looking at our KRAS inducible line mixed together with fibroblasts and fibrin, and over the course of a week, these endothelial cells and fibroblasts develop a complicated microvascular network that's perfused. As you can see here, when we use just wild-type cells, there's a really nice network that forms over the course of a week. We mixed in either 10% or 25% KRAS mutant cells, and this was really to mimic what we see in patients, which is a variable number of mutant cells within the nidus. What we found is that these mutant KRAS cells often were really preferentially found at these branch points. This really fits nicely with their angiogenic phenotype. And we also found that vessels where KRAS mutant cells were residing had larger lumens. And this again fits with some of the cell size phenotypes that we saw in 2D culture. And one of the nice things that you can do with these microfluidics devices is actually look at the permeability of the network. And you can perfuse in rhodamine, and in vehicle-treated cells, cell networks, you can see the leaching out of the fluorescence into the surrounding matrix. MEK, a PI3 kinase inhibitor, had no effect, so you still have a leaky vasculature. but the MEK inhibitor largely prevented leak. We also found that this was associated with reestablishment of V cadherin at junctions. And this is just shown here. So these systems are really quite nice at looking at mechanisms and also screening for pharmaceutical drugs that might alleviate phenotype. In work that was led by Josh Wythe at Baylor, we wanted to understand if KRAS mutations alone were sufficient for brain AVMs. And he used a CNS-specific KRI to delete a stop codon that would then drive G12D KRAS from the endogenous KRAS locus. And inducing at P1, he then looked in the adult for brain AVMs. And he found that in about 50% of the animals, there were very striking brain AVMs. So this is what a normal vascular network looks like. It's a nice hierarchical network. Within the animals expressing KRAS within the CNS endothelium, you could see evidence of direct connections between arteries and veins, these expanded lumens, and very tortuous vessels. And this is really very reminiscent of what you would see in a human AVM. Josh also showed that expression of mutant KRAS in an adult in a quiescent vasculature could also lead to brain AVMs. So this does not require necessarily a developmental or an angiogenic window. KRAS alone can drive the remodeling of quiescent vasculature. In our lab, we developed a zebrafish model where we used an endothelial-specific promoter to drive expression of either wild type or different versions of mutant KRAS. This was injected at the one cell stage, and this results in a mosaic embryo where some vessels will have the transgene and others won't. We found that expression of wild type KRAS didn't change vascular morphology, but expression of the mutant KRAS had a very dramatic effect on vascular morphology. A couple of the phenotypes are shown in this slide, where you can see ectopic sprouting. And we saw these very malformed vessels that had large lumens. They had what looked like lamellipodial protrusions. And these you can see within the intrasemitic vessel as well as within the dorsal aorta. Now, this vessel enlargement phenotype was really quite striking, and you can see this where there's M scarlet KRAS G12V expressing cells. The lumen is larger, and you can see that it has enhanced blood flow, as you can see in this bright field image. These vessels also have these odd lamellipodial protrusions. This is largely a cell autonomous effect because other regions of the same vessel or adjoining vessels had normal vessel diameters. And that's quantified here, where you can see that vessel segments with KRAS mutant cells had very large diameters. And because we had a nuclear GFP, we were able to quantify the number of cells in these vessels. And we wanted to know if the vessel enlargement was because of more cells or larger cells. What we found is that the number of nuclei per intrasemitic vessel was only had a very small change, but the area of the cells was greatly increased. And this again matches with what we've seen in cultured cells in vitro. We also found quite commonly these direct connections between arteries and veins. You can see here where GFP mutant KRAS cells are located, there's a shunt forming between arteries and veins, and this was again quite common in about 50% of embryos. We could also see this phenotype within the brain, as you can see here, enlarged vessels and abnormal connections between arteries and veins. We next wanted to understand if we could inhibit some of these phenotypes by blocking signaling downstream of KRAS, focusing primarily on the MEK pathway. So you can see here an example of this ectopic sprouting phenotype where mutant KRAS cells have more filopodia and ectopic sprouts. Exposing these embryos to vehicle control, DMSO, these vessels still have ectopic sprouting 1 day later, but in embryos, again, that have this enhanced sprouting phenotype, if we expose them to a MEK inhibitor, you could largely reverse the ectopic sprouting phenotype. We also found that using a MEK inhibitor, we could reduce the number of hemorrhages that we saw in the brain, as you can see quantified here. And finally, we wanted to determine whether we could actually reverse AV shunts. So we took embryos that had an AV shunt at 36 hours post-fertilization, and we treated those either with vehicle or a MEK inhibitor or a PI3 kinase inhibitor, and looked the next day at phenotypes. You can see here with vehicle control that the shunt is still present, but with a MEK inhibitor, we saw examples of embryos where this was resolved. And quantifying this, we saw about 75% of the embryos had their shunts resolved, and in comparison, a PI3 kinase inhibitor had no effect. Now, what's been really quite remarkable is really over the course of just several years, we've gone from really not understanding at all what the genetic cause of these malformations are to already translating this into the clinic. And there's a number of groups that are treating patients that have AVMs, that have mutations in KRAS or other MEK pathways with MEK inhibitors. And this is a case report of a patient with Cobb syndrome, where they have a spinal AVM as well as a thoracic AVM with evidence of KRAS mutation. And they treated this individual with trametinib, a MEK inhibitor, for several years. And you can see on angiography that the flow through the AVM is greatly reduced, suggesting that MEK inhibitors have some efficacy in patients with KRAS mutant AVMs. Now, in the last couple of minutes, I wanted to highlight some ongoing work where we're trying to better understand at the single cell level what is happening to the vasculature in disease. This is work that is driven by Thomas Walchley and Yvonne Radovanovic. They have done single cell RNA sequencing during development, as well as in the adult, looking at control brains from the temporal lobe, brain vascular malformations, and brain tumors. And they've sorted endothelial cells or looked at unsorted endothelial cell and surrounding perivascular cells, and sequenced more than 600,000 cells to try and understand what's happening at the cellular level, and importantly, looking at commonalities between vascular malformations and other vascular disorders such as occurs in brain tumors. What they found is that the pathological endothelial cells in both vascular malformations and in tumors lose their CNS properties. In particular, they lose blood-brain barrier markers, and they gain a peripheral signature, including MHC class II molecule expression. There's also disruption of normal AV specification programs, and quite interestingly, this reactivation of a fetal gene program. There's also strong activation of angiogenic and inflammatory signatures, and alterations in endothelial cell perivascular cell ligand interactions. And so I think a major question is what is KRAS contributing to these phenotypes? I've shown you examples of how KRAS can drive angiogenic programs. George Davis's group has shown that KRAS can disrupt endothelial cell perivascular communication, but a lot of these other phenotypes, we don't know how KRAS might be influencing them. So I'll just conclude and summarize what I've shown you today. Somatic endothelial KRAS mutations occur in the majority of patients with sporadic brain AVMs, and this mutation is sufficient for brain AVMs. We've shown that this is accompanied by ectopic sprouting, enlarged lumens and AV shunts at the earliest stages of AVM formation, and that inhibition of MEK can block and maybe even reverse AVMs. The molecular and cellular mechanisms are still being uncovered, but there's still a lot of work to be done there. We found that there's alterations in angiogenic program, in barrier integrity, in the extracellular matrix, and cell size. With that, I'll thank the people that have done this work. Emily Boudreau has done all of the zebrafish work. Rulin Wu and Nagar Khosraviani are working on molecular mechanisms of KRAS. And this work has been a really fun collaboration with a number of groups, including Josh Wythe, Yvonne Radovanovic, and Sarah Vasconcelos. And with that, I'll take questions.
Really beautiful talk, Jason. Thank you so much. So we do have a few questions, and so I'll just kind of read them off for you, and we'll go until we run out of time. So the first question is, do these bypasses only form between arteries and veins, or are there also artery shunts?
Yeah, so we haven't looked at that in particular. I think within the brain, we see a lot of, we initially characterize within the axial vasculature because it's the easiest sort of vasculature to understand, and there's clearly shunts between the artery and vein that occur very often. In the brain, we do actually see a lot of abnormal connections, and some of those do appear to be within one artery to another part of an artery, and then we do see artery-vein connections. We haven't quantified that as well in the brain vasculature to see exactly which vascular structures are being connected. But I think what we're seeing is this ectopic sprouting phenotype, and if we look early on, we see a lot of ectopic sprouting that's happening in the brain and in the trunk. And I think those connections, depending on where they land, they can actually, they're forming abnormal connections between vessels. We haven't sort of followed them long term to see what happens to some of those vessels, like particularly in the brain, whether some may be resolved. We do see that in the trunk sometimes. Some of those shunts do resolve on their own in a low percentage.
And do you know when you see these ectopic sprouts starting in let's say the ISVs of the fish, is there a preference for arteries and veins? Like, does it matter where the KRAS lands? Are you more, is there a preference there from where these ectopic sprouts start?
Yeah, we've been meaning to really quantify that. I think what we've seen, but we haven't quantified it, but it seems like the phenotypes are more prevalent, I would say, in the veins than the arteries. But we do need to really quantify that.
Mm-hmm. Very cool. And then maybe one last question here before we sign off is, most vascular networks have types of AV shunts in them, some occurring naturally on purpose. Do those areas are those rich in KRAS under like normal conditions?
Yeah, we haven't looked at that in particular. I mean, one of the questions that we have from our mouse model, we express mutant KRAS in the entire CNS vasculature, but the AVMs are quite localized. Mm-hmm. So I don't know, we're driving expression from the endogenous KRAS locus. So that would reflect potentially where KRAS is more expressed than other regions, but we haven't looked at that carefully to see if there's a lot of heterogeneity of KRAS expression level across the vasculature.
Very cool. Well, with that, it's right at 1 o'clock. So any other questions that you guys have, please feel to drop them in the questions box, and we will email them directly to Jason and Dionna. And so with that, I hope everybody enjoyed today's webinar. I know I did. And please look for more information about future events on the calendar on the NAVBO website, such as our DEI roundtable on immigration and visa issues on Tuesday, March 28th at 1 p.m. and upcoming webinars and symposium. So thank you everyone for joining us today, and we'll see you next month.
All right, thanks.